Hi everyone, this is Maria, and I'm going to talk a little bit about terms that you would use when we're talking about inheritance and different genetics. So the first thing we're going to talk about is Gregor Mendel. Now, Gregor Mendel is often referred to as the father of genetics, and he actually did his experiments way back in the 1850s. He was a friar who later became an abbot, and this is actually his monastery right here. And he was interested in studying how different characteristics were passed on from generation to generation. Okay, but what most people tend to know him for is he's the one who did all those experiments with pea plants. So this is actually his pea plant garden here. And this is actually just the more modern picture of it today, but pretty small, right? He also previously had used mice, and he also used bumblebees at different points in his life. Just sort of interesting to keep in mind. There's a lot of different terms when we're talking about the experiments that he did, so I'm going to try my best to break it down for you when going through. So the word characteristic is one of the things that we'll be using a fair amount. It's something that'll be inherited by the following generation. So it would just be flower color or height or seed form or pod form. There's a bunch of different ones when going through. Trait, though, is, I would say, we're probably going to use this term even more than characteristic. Traits are really, like, if we're being specific about it, it's a variation of a characteristic. So if I was talking about flower color as the characteristic, the traits would be white flowers and purple flowers. That kind of brings me to my next part that we have to talk about before we go on. It's these two different techniques of how Mendel actually did this experiment. And I'm just going to have it in the briefest of terms. So there's self-fertilization and cross-fertilization. Self-fertilization is if I took an egg and a sperm cell from the same pea plant and then combined them. A cross-fertilization would be if I took an egg cell from one pea plant and a sperm cell from a different pea plant and then combined those. We're going to end up shortening cross-fertilization as a term just to cross. And we'll often just use it in the sentence something along the lines of where, oh, the white flower plant was crossed with the purple flower plant, things like that. We're going to use that a lot of times when talking about cross-fertilization. For both self-fertilization and cross-fertilization, it's actually fairly easy to do with pea plants. You don't need a lot of complicated equipment. You really just need a paintbrush and a pair of scissors just to be able to trim off any of the parts of the pea flower that you don't want to either self-fertilize when going through. This next term is one that I know is a little bit tricky for students. So true breeding varieties. All it is a variety that'll produce offspring identical to the parent if self-fertilization occurs. So really what that ends up boiling down to is it's an individual that has identical copies of a gene variation. So that means that really if I'm using the letters if I'm using the letter A, and using big or little to talk about the different variations of a gene, that would be where a true breeding variety would be if I had big A, or little a, little a. So sort of as you see over here, where no matter what, like as long as we're self-fertilizing when going through, you're going to see that this is still genetically identical to the parental generation when going through. We'll also use the term hybrids, and that's really just if you have the offspring of two different varieties. So we'll talk more about that kind of as we go through. The last terms that are specifically related to Mendel's experiments are the P generation, which is just the same thing as saying the parental generation. You'll notice that usually if we're talking about the P generation, we're talking about a true breeding plant. Well, with Mendel's experiments, we're always talking about true breeding plants. There's the F1 generation, which is the result of crossing the two different true breeding P plants in the parental generation. The F part stands for filial, which is just Latin for son. That's not a huge deal. You don't need to know that, but just thought I'd throw it in. And then the F2 generation comes from crossing the F1 generation with itself. So in this particular case, this is where we would have use self-fertilization just on the F1 generation to get the F2 generation. We're not really going to go any farther from that. We might talk about the F3 or F4, but we're really going to stick with P generation, F1, F2. Those are really the ones that we'll talk about the most. All right, this brings us to a really important thing to know at this point. It's genotype versus phenotype. So when we're talking about genotype in class, we're talking about the genetic makeup. The genetic makeup is always going to be represented by letters. And I know I'm going to go back to the A that I keep on using for this particular one. So if you look at the eggs at the bottom, you'll see that there's big A under the speckled eggs right here. And then there's little a under the not speckled eggs right here. So remember that the big A, and the little a, those would be my genotype when going through. Now the phenotype is the physical trait expressed from the genotype itself. So it's really represented by the description of the trait itself. So when I was describing these eggs as speckled, well, that would be the phenotype. When I was describing these eggs as not speckled, that would also be the phenotype. It just depends on what we're looking at when going through. You're going to get a lot more used to both genotype and phenotype, but I always kind of remember it as genotype sounds like gene, so you can remember the genetic makeup. Phenotype, well, it starts with a PH, like physical, so then you can remember it that way. It matches up nicely. So as I was saying earlier, Mendel came up with a lot of great ideas that we actually still really use today. So I'm going to go through and talk about his 4 hypotheses. So let me start with this first one. There are alternative versions of genes that account for variations in inherited characteristics. So, for example, that kind of comes back to the idea of those different traits that we were talking about before. So if I'm talking about, for example, there are pea plants that could be tall and there are pea plants that could be short. Well, it'd be represented by the same gene, but there's slight differences within that gene that would cause this one to be tall and this one to be short. When we're talking about alleles, that's really what the term that we use. Alleles are just versions of genes. So if I was talking about the allele for this plant height, well, maybe I would say that for tall ones, it's a capital T would be one version of the allele. And then maybe for the short one, it would be a lowercase t. We're going to talk about what the capital versus the lowercase letters are when we're saying that, when going through. I'm going to talk more about that later. But just keep that in mind when going through alleles. There are versions of genes that still contain essentially the same information, just with a slight twist. Okay, so the second hypothesis that Mendel came up with was for each character or characteristic, an organism ends up inheriting 2 alleles, one from each parent. So this then sometimes you can inherit the same allele from each parent, or you could inherit one version of the gene from one parent and one version of the gene from another parent. So this is a great figure at the bottom of the page here. So if we're talking about homozygous versus heterozygous, homozygous would be, in this particular case, we have B's. So homozygous could be big B, or little b. The heterozygous version would be big B, little b. You could also write it as little b, big B. That's fine. But you'll notice that we tend to have a tendency to put the capital letter first, the big letter first, and then the lowercase letter. All right. Then Mendel's third hypothesis was about the idea of if there two alleles of an inherited pair differ, so essentially if there's a heterozygote, that's another way of saying if I have two different alleles, then one determines the organism's appearance and the other has no noticeable effect on the appearance. So really what that ends up boiling down to is this term of dominant allele versus recessive allele. The dominant allele will be the trait that will be expressed if present. So that's what I was talking about earlier with the uppercase letter. That's how it's represented in the genotype. The recessive allele is the trait that's only going to be expressed if a dominant allele is absent. So what it is, the lowercase letter. So if I'm looking at this fruit fly example right here, you can see that the one on the left has red eyes and the one on the right has white eyes. Well, let's say that the red eyes is dominant. Well, if the red eyes is dominant, then that means that if I had either a homozygous dominant case of big R, or if I had a heterozygote of big R, little r, I would have the phenotype of red eyes. If I was looking at the homozygous recessive of little r, well, that would leave me with the phenotype of white eyes for the fly. So just something to keep your eye on. You're going to talk about this a lot more in class as well, but just kind of keep this in your mind. All right, this last one kind of it has like an actual official name. So what it's called is the law of segregation. And so what that kind of boils down to is it's a sperm or an egg carries only one allele for each inherited characteristic, because allele pairs separate from each other during the production of egg or sperm. Here's all that's saying. If we're talking about these gamete cells, and that's what we're going to call sperm or egg cells, gametes, during meiosis, you're going to end up having those different alleles split into different ones. So if we're looking at here in this picture, we've got the red and we've got the white or gray. So if we're looking at, we've got red and white, and in a normal cell, you would have 2 copies of each of the red, and you would have 2 copies of each of the gray. And then you would end up going through like some interphase, and you produce your copies and things like that. But then you go through meiosis. At the end, if I'm looking at what's happening here, I see that each copy, we could say this could be either an egg cell or a sperm cell. It doesn't really matter. At the end, I only see actually 1 copy of the red and 1 copy of the gray. We've already talked about what this is, so hopefully you remember if you see that little bit, oh look, there's a little bit of gray on the red one. But keep that in mind, because this is actually a fairly important thing to think about. We're going to talk about, in the next few days, we'll be talking about how these hypotheses have changed and what information that we can actually add to them. But other than that, make sure you have all these terms down and write down any questions for your teacher, and we'll talk about it in class. Have a good night.